In this lecture, we're going to talk about microscopes. So units of measurement is really important. Microorganisms range in size from viruses, which are measured in nanometers. And a nanometer, if you look at the scale down here, that's actually a billionth of a meter. So it's a really small measurement. And then protists, which are eukaryotic microorganisms and are larger, they're measured in micrometers. And micrometers are a millionth of a meter. And at home, if you want to click on this link, it's actually really fun to look at. It actually shows you the size of different microorganisms and gives you an idea of the scale. So that's really good to look at home. There's three different types of microscopes that we're going to be looking at. Light microscopes are the ones that you've used in labs before, and the illumination source that's going to shine on the specimen is obviously going to be light. And electron microscopes, which there's a picture of one of those down here in the middle. The source of illumination that's going to shine on the specimen would be a beam of electrons. And then the third type of microscope that we have are called scanning probe microscopes. And there's a picture over here on the far right of these type of microscopes. And so they have a probe that will trace the surface of the specimen, and that will go to a computer, and the computer will then generate a picture of the specimen. We're going to be focusing first on the light microscope, and there's actually many different types of light microscopes. The type that you're used to using is called the Brightfield Microscope, but there's a lot of other types of microscopes that also use light as the illumination source. So we'll also look at the dark field microscopes and on down to the confocal microscopes. All of the light microscopes that we use today are called compound microscopes, and compound microscopes are defined as having two or more lenses. So you can see on this Brightfield microscope here, it is a compound microscope because it has two or more lenses. You have the ocular lens, and then you have the objective lens. So the light will shine up from the bottom, go through the specimen, go through the objective lens first, and then second will go through the ocular lens and then into your eye. So when light travels through air and then goes through the glass of the lens, it's going to be refracted or bent. And we talk about the refractive index, which is a measure of how much the substance is going to slow the speed of light as it travels through it. So air has a refractive index around 1. And then if you take a look at glass, glass has a higher refractive index because it's going to slow the speed of light more than air does. So when the light beam hits the glass, going from the air to the glass, it will slow, and then it's going to be refracted or bent. And then when the light beam goes from the glass back into the air, it will speed up, and then it will bend again. And so that's refraction. And you can actually see that. We've all looked at things sitting like in a glass of water, and you can see how the image is bent, and that's because of the different refractive index of the water versus the air. And you've also got some glass here too. So the lenses are made out of glass, which obviously have a different refractive index than the air. So it's going to slow the speed of light that's going through the glass from the air, and that'll refract or bend the light as it goes through. And when the light goes through the glass lens and refracts or bends, it's going to meet on the other side and will form an image at the focal point. And that will magnify the image, and then we can see a lot more detail that way. So just a couple of terms here. So we've got the focal point, which is where the image is going to be formed on the other side of the lens here. And then the focal length is the distance between the center of the lens and where the focal point is. Okay, so that is the focal length there. And the shorter the focal length, the higher the magnification. So the more curved the lens is, so the thicker it ends up being, the more it's going to refract or bend the light. And so your focal point is going to end up being closer to the lens here, and so your focal length is going to be much shorter on this lens that's on the left than it is on the other lens that was on the right. So the lens on the left, having a shorter focal length, is going to have a much higher magnification than this lens that is over here on the right that has a longer focal length. And unfortunately, we're not in the lab because everything's online right now. But if you were in the lab and you were to feel those objective lenses that you can switch, and you have like the 10x and 40x and 100x, if you were to feel those lenses with your finger, you would notice that the 100x objective lens is a lot more curved than like the 10x lens. And so it has a much shorter focal length on that 100x lens than you would on the 10x lens. So the 100x lens has a much shorter focal length and would have a much higher magnification. So that would be important to remember. Might be a good test. The Brightfield microscope that you use in lab is called that because the light, when it shines up from the bottom through the specimen, creates a dark image of the specimen against a bright background. So the background being bright, that gives the name Brightfield microscope. And the Brightfield microscope is a compound microscope having the two lenses, the ocular lens and the objective lens. To figure out the total magnification of the image, you just take the magnification of the ocular lens and then multiply that by the objective lens magnification. So it's pretty easy to calculate. So for example, if the ocular lens magnification, which you can't change, that's just what it's set at in the microscope. So let's say that is at 10x for the ocular lens. And then you're just going to multiply that by whatever the objective lens is set at. So maybe you're at 40x on the objective lens. So your total magnification of the image that you're looking at would be 400x. So the image that you're looking at is 400 times larger than the actual specimen. And then just another example, let's say that instead maybe you're looking at the image on the 100x objective lens, and so your total magnification would be 1000x instead. So that would be good to know for the exam, being able to figure out the total magnification. So if I give you the ocular lens and objective lens magnification, could you figure out the total magnification? Okay, and this is just showing you the two lenses, just showing you the light shining out through the bottom, through the specimen, and then how it goes to the objective lens, and then the ocular lens, and then finally into your eye. And you can see how the light will be bent and refracted through both the objective and the ocular lens, and that'll magnify the image. Okay, we have three very important terms that we use with microscopes. So make sure that you know these three terms for the quiz and the exam. We have magnification, contrast, and resolution. So magnification is pretty easy. So it's just how much bigger the image is compared to the actual size of the specimen. So here's a picture of magnification. So on the right here, we've got the actual specimen. This is a root tip of a plant, and this is its actual size. And here we've magnified it twice the size, so we've got 2x as our magnification. So it's twice as big as the actual specimen. So that would be magnification. That one's pretty easy to understand. And then the next concept is contrast. So contrast is just how well different parts stand out. So looking at a cell that has not been stained under a Brightfield microscope, you can see that this cell, it's really hard to pick out the parts of the cell. Like, it's really hard to see exactly where the nucleus is. So for example, the nucleus is actually right here, but it's pretty hard to pick that out because there's not a lot of contrast. So you really can't see the differences between the parts in the cell, and so things don't really stand out. But if you stain the cell, that increases the contrast and allows you to pick the different parts out of the cell, and so now you can really see the contrast with the nucleus between the other parts of the cell here. Okay, so that's contrast. Okay, and then resolution. This is talking about how sharp or clear the image is. And it's just defined as the ability of the lens to distinguish between two objects that are close together. Okay, so like if I hold up two markers and maybe I'm standing, right in front of you, and I've got two markers that I'm holding up. You could tell that I have two markers that I'm holding up. But if I stand a half mile away from you and I'm holding up two markers, it would be really hard for you to tell that I'm holding up two markers because you'd have, much lower resolution, and it would be hard for you to tell that, distinguish that I've got two markers that I'm holding up. So there's a couple things that affect how sharp or clear the image is that we see in our microscope. The wavelength of light that we use affects the resolution. So the shorter the wavelength of the light that we use, the higher the resolution. So with the visible light that we use, the blue and violet wavelengths of light, that'll give you higher resolution. And then also with our microscope, the larger the aperture of the lens, the higher the resolution as well. And we'll get to what the aperture of the lens is in just a minute. Okay, but those will be good things to know for the exam, knowing that a shorter wavelength of light gives you a higher resolution, and also a larger aperture of the lens will also give you higher resolution. So you get a much sharper, more detailed image with those things. Okay, so let's just show you some pictures of resolution. All right, so we've got our plant root tip here, and we've got the same magnification on both of these, but if you look on the bottom, we have very different resolution. So you can see on the picture on the left, we have poor resolution there. So you can see it's just kind of blurry, and it's hard to pick out the details. It's not a very sharp image at all. But the picture on the right has much higher resolution. It's more sharp and detailed image. So you can see these little, dots in here and these little, hairy things sticking out much more clearly. And so this has a much higher resolution than the picture on the left. Okay, so what is the numerical aperture of a lens? So this is defined as the ability of a lens to gather light. So the lens on the left has a smaller numerical aperture. It's not able to gather as much light that is shining up through the specimen from the bottom. It's not able to gather as much light that's shining through the specimen. And if you compare that to the lens on the right, which has a higher numerical aperture, you can see that it's able to gather a wider angle of light that's coming up from the specimen from the bottom there. And so by having a wider angle of light that it can gather more light and have it more separated, and so you can see a lot more detail when the numerical aperture of the lens is a lot higher like that. So if you have this greater angle like that, able to gather more light, then you've got higher resolution and you can see more detail in the image. So with our objective lenses, the lenses that have a higher numerical aperture will be the objective lenses that have a higher power. So like your 100x objective lens, that has the highest numerical aperture on your Brightfield microscopes. And the higher the numerical aperture, the shorter the working distance, so the closer it's going to be to the microscope slide. So, and you guys probably all remember that from working with these in lab. The higher the power on the objective lens, the closer the lens got to the microscope slide. So, and that also means that you've got a higher numerical aperture as well, and also a higher resolution, and you can see more detail and a clearer picture. Okay, so with these 100x objective lenses on our Brightfield microscopes, we do use immersion oil with these, and that's because it increases the resolution. And so if you do try to use the 100x lens and you forget to use the immersion oil, your picture will be very blurry and you have very low resolution. The reason that is if there's just air there between the microscope slide and the 100x objective lens, the air has a different refractive index. And so when the light goes from the glass, hits the air, it's going to refract and bend. And a lot of that light that went from the specimen will then miss the objective lens and won't make it into the objective lens. And so you're going to miss all that detail. And so then the image that you're seeing is not going to be very sharp or detailed. But if you use the immersion oil, the oil has a very similar refractive index to the glass. And so when the light goes from the glass, hits the oil, it's not going to bend. And so it just goes straight into the objective lens, and that will form part of your image. And so you can see all of that detail in your picture, and that'll give you much higher resolution. Okay, so because the oil and the glass have a very similar refractive index, the light rays aren't going to bend, and that's going to allow it to go right into your lens, and that'll give you that detail of the picture, and that'll increase your resolution. Okay, whereas if you forget the oil and you just have the air there, the air has a different refractive index, the light will bend, and then the light rays don't go into the lens and you miss all that detail from your image, and that'll give you a lower resolution. So that's why you want to use the oil with the 100x objective lens. Okay, and then this table you don't have to memorize, but it's just good to take a look at. And this is just looking at the different objective lens power going from the 4x up to the 100x lenses. And you just need to realize what's going on. So remember we talked about the numerical aperture of the lens. So that's just the ability of the lens to gather light. And remember that was important for resolution. So the higher the numerical aperture, the higher the resolution, the more sharp or clear your picture is going to be, the more detail you're going to see in your image. So you can see the 4x, very low numerical aperture, but if you get up to the 100x, you have a much higher numerical aperture, so you're going to have a much higher resolution, much more detailed, sharp, clear image on the 100x power. And then remember the focal length. That's just the distance between the middle of the lens and where the focal point is, where the image is formed. Remember, the shorter the focal length, the higher the magnification. And you can see here that the 4x has a pretty long focal length, and when you get up to the 100x, that has a very short focal length. So the shorter the focal length, the higher the magnification, which makes sense on the 100x. And then the working distance, remember that's the distance between the lens and the microscope slide. And we all know that the higher the power on the objective lens, the closer it gets to the microscope slide. And so you can see that here, 100x is very close to the microscope slide as a very short working distance. And then here's the resolution, how far apart things need to be. So you can see them as two separate objects, and you can pick out that detail. And so you can see on the 100x lens, things have to be about 0.2 micrometers apart for them to be seen as separate objects. And if they're closer together than 0.2 micrometers, then they blur together and you can't pick them out as being two separate objects. So the maximum resolution for our Brightfield microscopes are about 0.2 micrometers, which is about 200 nanometers. So that's about the size of a bacterial cell. Okay, and anything smaller than that, we can't resolve. It just kind of all blurs together and we can't pick out that detail. So we can't see most viruses with a Brightfield microscope. Okay, they're much smaller than 200 nanometers. Okay, so we're going to need an electron microscope to see most of our viruses. Okay, there are some exceptions. There are some very large viruses that you can see with a Brightfield microscope, but for most of them, you do need an electron microscope to see them.